Good morning everyone, good morning. Good morning Akshay, how are you doing? Good morning everyone. Okay, so friends, in the yesterday class we were discussing about this particular question number 38, is it? I hope every one of you remember the answer for the question number 38. What's the question asked, friends? A very direct, very straightforward question. Good morning Vinita. Which one of the following statements with respect to the development in amphibians is correct? As we have very clearly discussed that, friends, the answer will be the gastrulation. We have discussed very clearly with the help of diagram. Good. So first, the process of gastrulation will start with the invagination of bottle cells. Then, friends, followed by the involution. At the end, it will undergo the process of epiboly. I hope every one of you remember that. Okay. Friends, let's start. Everyone start the question number 39. I want every student has to answer this question very correctly. Okay. Look into this. Saima, Priya, Akshay, Shafia. We have directly discussed this question in the class. Okay. Very straightforward question which we have discussed, in the PPT itself. The question is little logical. Think it and give me the answer. We studied it very clearly. Think it, no problem. Good morning, Sushma. Good morning. Yes. Please give me the answer for question number 39. Okay. Anyone else? Abdul has given 3. Okay. Look into the question very carefully. It's an easy question, straightforward question. Even it is not from the depth of the question, depth of the concept also. Very straightforward question. Sumaya said B. Look into this, Sumaya. Look into this, dear, very correctly. During normal development of the sea urchin, beta catenin accumulates predominantly in the micromeates, which are fated to become endoderm as well as the mesoderm if GSK3 is blocked in the developing embryo. First of all, what is the function of the GSK3? We all people know that. GSK3 is the one which will block, keep it in your mind, which will blocks the beta catenin. Correct now, friends? We all people know it about it very well. And friends, what is the function of this particular beta catenin? Beta catenin is the one which is basically involved in the formation of the endoderm as well as in the mesoderm in almost all the blastula cells. Now you only tell me, the question is very simple. If GSK3 is blocked, which means that GSK3 is not acting, the GSK3 get inhibited. Please remember, friends, when the GSK3 get inhibited, what is going to happen, friends? Beta catenin get activated. The moment beta catenin get activated, what happened? Beta catenin entered into the nucleus of all the cells. Which means you have to follow me correctly. Beta catenin enter into the dorsal cells also. Beta catenin can enter into the ventral cells also, and it will cause the development of the endoderm as well as the mesoderm. Hence the correct answer will be the C. Now option B, beta catenin will accumulate in the nuclei of all blastula cells leading to an ectoderm ball. Not only ectoderm, it is also required for the formation of the it is required only for the formation of endo and mesoderm. The beta catenin do not have any direct role or indirect role in the formation of a ectoderm. So option B is wrong. Option A is also wrong, because friends, they said that they have given the ectodermal. So please keep it in your mind, ectoderm cannot be formed by the beta catenin, only endoderm and mesoderm. Hence undoubtedly C will be the correct answer. And beta catenin which accumulate in the nuclei of large micromeres. Option D. No, that is also wrong. Sushma, why this option D is wrong? Because the beta catenin not specifically entering into the nucleus of the large micromere. No, because the GSK3 is inhibited. So what is happening? The beta catenin is going to enter into the almost all the blastula cells. Every single blastula cell it will enter, not specifically to the micromere, not specifically to the macromere. Hence my friends, the correct answer and the specific answer will be the C. Now, can anyone give me the answer? Here we have discussed that in the question, if GSK3 is blocked, that is a question. Now friends, how does this particular GSK3 is blocked? First of all, I want you people to remember without fail, GSK3 is blocked with the help of a lithium chloride. This is the unique statement I want you to remember. GSK3 is blocked by what, my friends? Lithium chloride. Don't get confused. Next time if they ask you a question, always remember that which can block the GSK3, which is the lithium chloride. Okay. Good morning Sanjay. Good morning Deepika. Deepika, without fail, see the recording of this one particular question, which is the question number 39. Okay, don't miss it. Okay, friends, I can tell you one clear-cut statement. Out of 13 units in the CSIR NET Life Sciences question paper, only the unit 5 part B questions are very direct question. I can tell it very clearly every time. Now whatever the paper that you are going to write, even in the 17th September also, the questions will be very direct. The questions will be very straightforward. There is no doubt about it. So that's why in case of this particular chapter of developmental biology, try to answer 2 to 3 part B questions without fail. Okay, now you can tell me just like that. In which of the following, good morning Annie, please continue. Annie, watch the recorded of yesterday, don't miss it. In which of the following stages of rhabdiosis embryogenesis do the visible distinction between the adaxial and abaxial tissues of the cotyledons become initially apparent? Saima, what will be the answer? Akshay, Shafia, what will be the answer for the question number 40? Think it. You know, just for your understanding, I am saying, friends, adaxial means upper side, abaxial means lower side. Okay, so friends, we all people know that adaxial side or upper side, what kind of mesophyll cells we can observe, friends? We can observe columnar palisade mesophyll cells. We can observe columnar palisade mesophyll cells. And friends, in the lower, what we can observe, friends, in the abaxial tissues, we can observe the spongy mesophyll cells. And friends, what will be in the middle? Middle to the adaxial and abaxial. Adaxial is the upper, lower is the abaxial. Then what will be the middle? Middle only, friends, we will be calling it as the torpedo stage, which you can observe cotyledons. Very simple, straightforward question. It's not at all difficult. Good. Question number 41. See friends, this question is just directly from the definition. I hope every student remember, when I am explaining the gas relation, I have explained you this particular experiment with the help of a diagram, and there also we have solved one question, and the question is directly from the experiment. Don't confuse, just think it. Take your time, think it. Slowly, Sushma, please don't be hurried here. Don't spend 20 to 30 seconds. Don't be hurry. You're all giving answers very fastly. That is not idealized. Okay, you know, this question is not a difficult question. This question is a logical question. It's not a difficult question at all. In a transplantation experiment, the area of presumptive ectoderm, see, imagine this is the presumptive ectoderm from an early frog gastrula was transplanted to a region of the newt. Newt means frog gastrula, destined to becomes the part of the mouth. See here, this presumptive ectoderm we have transplanted to a particular part in the frog which is going to become mouth actually. Now the resulting salamander larva had a frog-like mouth parts instead of balancers as observed during the development of a wild type newt embryo. This is an example of. So my friends, please listen, don't get confused with my words. Anyway, it is not determined. It's not the process of determined. What do you mean by the word autonomous specification? Autonomous specification means on its own it is getting developed into whatever it wants to develop. So it cannot be an autonomous. So someone is getting, someone is getting influenced, but you have to remember whether it is getting influenced regionally or whether it is getting influenced genetically. You people has to remember. Don't confuse with my words. Follow me. The presumptive ectoderm having the genetic tendency, the presumptive ectoderm having the genetic tendency or the genetic specificity or the genetic ability to modify any tissue or to decide the fate of any tissue. Now, why the answer cannot be a regional specificity of interaction? I'll give you the valid explanation for this point. Every student pay attention. Why it cannot be a regional specificity? Because friends, regional specificity means, suppose example, this presumptive ectoderm, instead of forming mouth, suppose example, it has formed suckers. It has formed suckers. Or instead of forming the mouth and suckers, it has formed some other balancer proteins or balancer genes. Now what we can say that this will become a regional specification, which means presumptive ectoderm can convert the targeted region into mouth also, can convert the targeted region into balancer also, it can convert the targeted region into the sucker also. But what has happened? The presumptive ectoderm can convert the targeted region only into mouth. It cannot convert the targeted region into the sucker. It cannot convert the targeted region into the balancer. Don't confuse. So what I want to say, the presumptive ectoderm, it is genetically specified. It is having a genetic tendency that it is having the ability to convert the region only into mouth. It cannot convert any region into any other part apart from the mouth. So it cannot be a regional specificity of interaction. It will become a genetic specificity of interaction. Don't worry at all. If you are not 100% sure about the question, please don't give the answer. The answer will be the B. Don't worry. The clear-cut explanation was given. Don't worry about it. You can read it. If you don't understand, you can ask me. Which of the following R gene products do not contain NBS LLR domain? You know, this is it's not a logical question. Most of the students won't read this. Even though you don't answer, that's okay, fine, no problem. You know what is NBS? Nucleotide binding site. That is called NBS. What is LRR? L represents, friends, leucine. Okay, leucine rich repeat. Correct, Sushma, correct. Try it, anyone can tell. Okay, friends, this question, even though you don't know the answer, that's okay, because it is a generalized question which you have to know it. First of all, which of the following R gene products do not contain NBS LRR domain? First of all, what are R genes? Here the R genes represent, friends, plant resistant genes. Okay, so friends, actually this plant resistant genes are present in the wheat also, as well as the sorghum also. It is present in many kind of a plant. Now please remember, this NBS, that is the nucleotide binding site and leucine rich regions, these domains are not present only in the MYO protein of barley. Rice also it is present, Arabidopsis thaliana it is present, tobacco also it is present, wheat also it is present, sorghum also it is present. The only domain which the only NBS LRR domain is not present is only in the barley. That's okay if you don't answer these questions. Now let's look into this. How many questions were asked in the unit 5? 38, 39, 40 and 41, then 42. There are 5 questions were asked in the unit 5. 5 into 10, 2 marks. 5 into 2, 10 marks. Now you have to remember that our goal is not to answer 5 questions. Our goal is answer to minimum 2, maximum 3 compulsory. Minimum 2 and maximum is 3. Don't go beyond that. Unless if you go beyond that, unnecessarily you will make the mistakes. So no need to worry about it. So friends, listen. We have started with question number 21 and we have ended with the question number 42. Total how many questions are there? 21 questions are there. So on average, from 1 to 5 chapters, we are getting 4 part B questions. So in your upcoming September exam, every student keep it in your mind. Out of this 21 questions, your target is answer 10 questions. Your major target, your minimum target. Your minimum target is answer 10 questions. When I use a word answer 10 questions, which means that you have to answer correctly. That is what it means. Minimum you can go for 10, maximum you can go for the 13 questions. If you are 100% sure about the 14th question, then only I will encourage you to go for that 14th question. Otherwise minimum 10, maximum 13. Because friends, we did not dependent on only this. We have still 7 chapters are there. Let's look into this. Okay, question number 43. Okay, which is a very direct question. Okay, Archana, try it. Everyone. Friends, they are asking a question where the ATP is consumed. They are asking consumed. Not synthesized, consumed. And please count yourself how many questions you made it wrong, how many questions you did correct, and why you made the question wrong. That is also important. Yes, yes. Saima, Annie, what is the answer? I don't know what you people are reading, huh? Friends, how come is this C? You people are reading this from last 6 months to 1 year. You're not getting a question. What's the question asked? Sumaya, what will be the answer? Shafia, Deepika, why you are making it wrong? I am not getting. The question asked is where the ATP is consumed. That is the question asked. Now you know, right, we will consume the ATP in the reduction reaction. Correct na? And we will consume the ATP in the regeneration reaction. Right? In the reduction reaction, in order to convert the G3P to the GA3P, how many ATP we will spend? We will spend 6 ATP. And in the regeneration, in order to convert the GA3P to what we can call RUBP, we will spend 3 ATP. Is it or not? We will spend 3 ATP. So we will spend total 9 ATP. We will consume at which step? Reduction step and regeneration step. I don't know. What do you mean by the word carboxylation? Carboxylation step, why we will spend the energy? That is absolutely wrong. Carboxylation means it is a kind of a breaking step. Right? We will only use in the reduction and regeneration. See, once again I am telling to all of you, you all people know the answer for this question. You all people read the photosynthesis 10 times and 15 times. But what is the use if you don't interpret the question correctly? Now I will tell you one word, listen to me. 90% of the students, they never qualify the exam. It is not because they are intelligent. It is only because they always fail. They will always fail in analyzing the question. Only 10% of the students, even though they are having a less skills, even though they did the less practice, they will analyze the question properly. That's what I am saying. That's what experience will come. That's what you have to analyze the question peacefully by spending 30 seconds. I will be worried more if you don't answer a simple questions. You will make simple questions wrong because of your poor thinking. That's what you have to change. Okay, slowly analyze the question. Don't worry, don't worry. Okay. Next, dirigent proteins predominantly play an important role in the biosynthesis of. Sometimes, you know, instead of giving the dirigent proteins, they will use the word DIR. We can call plant dirigent proteins. Correct, Tanvi. ATP synthesized in the carboxylation. Wonderful. It's okay. Remember, you might not read this, but remember, dirigent proteins are exclusively required for the biosynthesis of lignins or the lignans. You remember, I told to all of you while explaining the secondary metabolites, I told to all of you that, friends, lignins can exhibit in the different forms. Correct na? I hope every one of you remember, we have discussed some type of a lignins. That is the condensed type of lignins also, and we discussed about the hydrolyzable also. And I also told you that, friends, lignins, please remember, friends, lignins are the most toughest molecules after the cellulose. Cellulose is the most toughest. After the cellulose, lignin is the most toughest. In the lignin biosynthesis, we will use this particular DIR, dirigent proteins. Plant dirigent proteins. Don't get confused at all. Now, friends, what are the examples of the dirigent proteins? I want you people to remember the dirigent proteins also. Okay. Regioselective dirigent proteins and, friends, enantioselective dirigent proteins. Enantioselective dirigent proteins. These two are the examples of the dirigent proteins which will be involved in the biosynthesis of this particular lignins. Don't worry, don't worry, don't worry. Direct question. Think and answer, think and answer. Which one of the following is used in the organification of the tyrosine residues in the thyroglobulin protein during thyroid hormone biosynthesis? If you are belonging to the zoology background, you might done the experiments also. Slowly, slowly, slowly. Yes, yes, yes. Okay, okay. What is the question they asked? Which one of the following is used in the organification of the tyrosine residue in the thyroglobulin protein? First of all, what is organification? Organification is a biochemical process which is happening in the thyroid gland. What we will do in the process of this particular organification or the biochemical process, what we will do, we will introduce the incorporation of the iodine into thyroglobulin. When I introduced the iodine into the thyroglobulin, what I am going to get, friends? I am going to get the thyroid hormone. Very simple step it is. If I want to produce the thyroid hormone, what should I do? I have to introduce the iodine into thyroglobulin. Now the point is very clear. What is the enzyme involved in this particular reaction? The enzyme involved in this particular reaction is a thyroid peroxidase enzyme. Once again, every student listen to me carefully. Thyroid peroxidase is the name of the enzyme which will introduce or incorporate the iodine into the thyroglobulin in order to produce the thyroid hormone. And what is the speciality of this particular thyroid peroxidase enzyme? It can incorporate the iodine into the thyroglobulin only whenever the iodine is in the oxidized form. If the iodine is in the reduced form, this enzyme is not specific for that reaction. If the thyroid peroxidase wants to be active, it can incorporate only oxidized iodine into the thyroglobulin. Please do not get confused with that particular point. Hence the correct answer will be C. Simple, straightforward, direct question. Okay. The dark current in retina is due to, you know friends, which we have discussed in the rhodopsin pathway, also in the cell signaling also, and they have asked with respect to the human physiology here. Okay, so friends, all the students remember all the time one common point every student has to remember. Very good, very good. Inflow of sodium, wonderful. Inflow of sodium we can call the dark current. Always remember, inflow of sodium we can call it as dark current. Outflow of sodium we can call it as the light. Correct na? And they are asking very clearly, the dark current in the retina is due to, the answer is very simple, opening of the sodium channels in the outer segment of the photoreceptor. And what is this pathway? Rhodopsin pathway. Please don't get confused with this point. Okay. Which one of the following is not present in the filtration slit of the diaphragm? Think it, friends. Think it, think it, think it. My friends, the question asked is which is not present. They're asking not present. This is not a logical question at all, but this information has to be remembered. Okay, in order to answer this kind of questions, at least see, listen my statements. If you want to answer this question, at least you people should know the functions of at least 2. If you don't know the functions of at least 2, you can't answer this question. Okay, don't worry. NPH1, remember, NPH1 is a protein which is present in the glomerular slit of kidney. Okay, NPH2, it is present almost all part of the kidney. It is present in every kidney slit. Every kidney slit it is present. And what about the nephrin, friends? It is present in the glomerular podocytes. Glomerular podocytes. So option A, option C, option D, these 3 are correct options, which are with respect, all the students remember, with respect to the filtration slit. Always remember that. Now the question you have to remember, friends, paxillin. Paxillin is an important protein. It is actually involved in the extracellular matrix protein. And this paxillin, friends, very specifically attaches to the focal adhesions present on the extracellular matrix. Don't get confused. It's okay, you don't know the answer, no problem. But remember now what is the function of every single protein. Don't get confused. Hence, which is not present in the filtration slit of the diaphragm is, my friends, paxillin. Don't get confused with that. This question I have given wrong. Okay, I will upload the question tomorrow. I have typed question wrongly here. Okay, yes. Now question number 49. Easy question, direct question. I have explained to all of you. I want every student, especially I am telling whomever attending my classes. See, Tanvi, the paxillin on the ECM, right? ECM contains what, dear? ECM contain collagen, correct na? ECM contain nidogen, ECM contain fibroin, correct na? So all, see, example, this is the collagen, this is the nidogen, this is the fibroin. To the collagen, nidogen, fibroin, elastin, whatever the ECM proteins are there, in those ECM proteins, this particular paxillin is present. Paxillin attaches to this particular ECM proteins with the help of an interaction or with the help of a force or a joining, which we can call focal adhesion. With the help of focal adhesion, paxillin attaches to almost all the ECM proteins, not specifically to one ECM, almost all, with the help of focal adhesion. Okay. See, I am telling all of you, Sumaya, Saima, everyone has to answer this question correctly. Priya, especially Deepika, everyone. We studied this. Akshay, Annie, you have to answer this. I have explained you many times, many times. At least you people remember that I have explained it. Allelic, non-allelic. Especially when we are doing the extension of a Mendelian principles, I have explained it to all of you. Extension of Mendelian principles, I have explained you this. And I have explained it the same example only. Otherwise, you check the PPT. Okay. Now Saima, Deepika, Sumaya, Shafia, please tell me, every student has to answer my question. Originally, which eyes will be found in the fruit flies? I am asking the wild type. Which wild type eyes? What are the eyes which are the wild type in the fruit flies, in the Drosophila melanogaster? Wild type eyes are which color, Annie? Saima, wild type eyes are which color? Wild type eyes are red color. Correct na? But what is this apricot? Every student pay attention. Apricot is a X-linked recessive mutation. Apricot is what? X-linked recessive mutation, which is basically producing what? Not the red color. What they will produce, friends? They will produce the light orange red color. Very good. Light orange color. Correct. Which is the light orange color. Now, I want you to know whether light orange or apricot, what kind of a test we will do, friends? We will do the test of complementation. Yes or no, friends? We will do the test of complementation. Now, what you have to remember, the gene is only one. Every student has to remember the gene is only one. But the gene, because of a mutation, I want every student has to listen to me carefully. The gene is only one, but the gene, just because of the mutation, one gene can code for one phenotype. We all people know that one gene can code for one phenotype. Because of the mutation, one gene is responsible for the two phenotypic characteristics, right? Then that is what exactly we can call the non-allelic. It cannot be allele. Pseudo-allele means what? False allele. That cannot be a false allele. And we cannot consider the option D even it is completely wrong. It cannot be a paralogous. Obviously, the answer will be the non-allelic, where one gene, because of the mutation, it is responsible for the two phenotypic characteristics. And this we can easily identify by the complementation test. Wonderful. Very good. Very good. Fantastic. If you don't know this, okay. Question ID 50. Here there is a simple formula, but try, every one of you try. Tanvi, can you tell me the formula for the penetrance? You know, I think when I explained you the penetrance, I have explained you one example of a Hawaii bird. I don't know whether you remember it or not. Okay, you try it, friends. You try it. No problem, try it. Take your time, but I want you to just give me an guess. Take your time. I hope every one of you know what is polydactyl. Having more number of toes or fingers. You might observe, right, some people having 6 fingers. They are having 2 thumbs. That is called as polydactyl. See, the question is difficult. Which is the correct? Okay, you have to choose the more appropriate. Okay, Sumaya, can you tell me, dear, why you have given the answer as B? I have explained it to all of you very clearly. Okay, now anyway, don't worry, just listen to me carefully. Penetrance means what I said to you. I hope every student remember penetrance and expressivity. I hope every one of you remember it very clearly, right? Penetrance means that I told to all of you that one genotype is exclusively coding for one phenotype. one genotype is going to code for the phenotype. Then what I can say, the expressivity will be the 100%. Once again, every student remember, there is one gene. Friends, one gene basically can code for what? One gene can code for one phenotype. So listen, follow me correctly, which means that the gene is working very effectively. I can say that the expressivity and penetrance is 100%. But what every student has to remember, how many genotypes are there? 42 are there. But my friends, what will be the expressivity? Only 38 is expressivity. 42 two genes are there, 38 is the expressivity, correct na? Then I never say that the expressivity will be the 100%. That is not correct, because you can see here, if one gene perfectly coded for one phenotype, the expressivity and penetrance is 100%. Here 42 genes are only coding for the 38, then it cannot be 100%. In order to estimate these particular options, you have to use sometimes a formula, but 99% you do not require the formula. But here this is an example of variable expressivity. No. The polydactyl showing complete penetrance. No. The option will be A and B will make you confused. During that time, I want you to use a small formula. Penetrance equal to phenotype by the genotype into 100. Right, this will be the formula. Even though you use this formula, you can get the correct answer. 38 by 42 into 100. So how much you will get? Nearly 90% of the penetrance you can get it. Okay, I mean to say expressivity we will get it. Now friends, penetrance cannot be the 90%. Okay, XC, because penetrance is 100%, there is no doubt about it, but the expressivity is how much? Expressivity will be the 90%. Hence the correct answer will be the B. Don't get confused that friends. Here penetrance they were very clearly given 42, but the expressivity is only 38, which means that there is a variation in the expressivity only. So what is that variation? 90%. Okay, we should not use the word estimated. Why we don't use the word estimated? Because the option will be confused, because they have given the word estimated. By using the penetrance formula of genotype by phenotype into 100, we can easily estimate that particular expressivity. So that's why we don't use the word estimated. You should not use the word estimated. Okay, hence the correct option will be the B. I hope everyone understand without confusion. Answer will be the B. Let me know if you have confusions. Okay, the direct question. The basic differences between the direct repair and base excision repair. You people studied n number of times. At least you know the what do you mean by the word direct repair. You could answer this question clearly. While explaining the DNA repair mechanism, every time I used to highlight one point about this direct repair. Every time I used to highlight the same point. At least if you remember that point, you can answer the question. I told you many times. Friends, when compared to all the repair mechanism, direct repair mechanism having a special quality. Take time, give me the correct answer. Slow friends, you're not understanding what I will say. Okay, think it. Okay. Actually, there is no doubt the option A is correct. There is no doubt about it. Okay. But this time they have given option B as the answer. Okay. But many people challenged this particular question also. But there is no doubt about it. The option A is correct, because we studied, friends, when compared to all the repair mechanisms, the direct repair mechanism is the repair mechanism where we don't remove any kind of a base. We don't remove the base, we don't alter it, we will just repair that without replacement. In the base excision repair, the DNA containing the distortion, suppose if the wrong base is involved, we will remove that and we will replace with the correct base. Obviously option A will be the correct. Okay, but I don't know why they have given the answer B, and many people challenged also. Okay, question number 52. You know, friends, let me tell you one point to all of you. Okay, question number 52, listen my words, listen to me. Question number 52, question number 53, question number 54, and question number 51, 52, 53, 54 and 55. If you people really study question number 51 to 55, if you people really study the NCERT, or if you are a teacher who is teaching to +1 and +2, trust my words, blindly you can get 10 marks blindly, because every single question came from the NCERT, exclusively 11th standard, exclusively 12th standard NCERT. If you are really a teacher of +1 +2 biology, or if you are thorough with the NCERT for this particular chapter, you could answer the questions without thinking. Okay, but what we people will do is, I am not saying that most of the students will leave the 9th chapter. That's okay, but if you want to get it, at least focus on the NCERT level, because every single time we'll get the NCERT. I was talking about the part B, and if you look into the part C also, you will get surprised. Every question is directly from NCERT. Okay. Answer: eukaryotes are classified into 5 to 6 super groups based on the phylogenetic studies. Which one of the following statement about the eukaryotic supergroups are false? False, they are asking. See, Tanvi, look into the question, dear. The question is, first of all, I am just giving one hint to all of you. Only this question is little difficult question. Only this question. They have asked this based on phylogenomic studies. When I use a word phylogenomic studies, every student has to remember one point: more closely related species. Okay, now friends, if you look into this particular fungi and animals, they have given a word that they are more closely related to each other than either group is to the plants. Of course, friends, if you consider the animal and plant, and if you consider the animal and fungi, we are very closely related. Why? Because all the students remember, fungi and animal, these both are belongs to a particular supergroup called as ophthioconts. So option A is absolutely right option. Amoeboja ophthicons are the unicorns. Obviously, friends, this is also right. This is a direct statement. Land plants or green algae belonging to the Archaeoptera. This is called supergroup. Obviously, right statement. Alveolites and amoeba belonging to the same supergroup. No, alveolites belongs to the different group. Amoeba belongs to the different group. Hence, the wrong option will be the D. Okay, it's okay. Even though you don't know the answer, leave this question. Now see how easy questions. Question number 53. Even though you remember your NCERT, you can answer. Mammals, we have discussed it 10 times. Correct, Saima. Answer is D. Okay, before question. 53, everyone answer 53. No need to think also. We studied more number of times. Others, Sumaya, Deepika, Annie, Priya, why you are not answering? Right or wrong answer? Friends, why you are thinking this much of time? How many times we have studied? Which is the rotational cleavage. How many times we have studied? Good. Okay. Question number 54. Same NCERT. This is from the pteridophyte, which is the 11th standard. Okay. 11th standard, there is a plant kingdom chapter. In that bryophytes, pteridophytes, gymnosperms, angiosperms are there. From that we got a direct question. Pteridophyte, you people no need to think about it even. It's a direct question. Which is the diploid sporophyte. To be honest, it is the lines of NCERT. Okay. So lines of NCERT. Okay. Diploid sporophyte. Very straightforward question. Okay. Next. Question number 55. Which one of the following gases is present in the stratosphere at a concentration higher than its concentration in the troposphere? We all people commonly know, right? Okay, so what will be the answer? Answer will be the ozone. You know now friends, what does that particular atmosphere contains? Friends, this kind of questions are very common in our part B. So that's why I want you to remember all the composition. Atmosphere contains mostly what? 78% is nitrogen. We all people know very well. And it contains 21% oxygen. We all people know that it contains 0.93% of the argon. Correct na? It contains 0.0% of the carbon dioxide. And friends, some trace amounts, very trace amounts of neon is present, helium is present, methane is present, argon is present, krypton is present. Correct na? These are all present at the very trace amount. And you know very well the atmosphere containing what friends? Two layers. Correct, right? So one we can basically call it as what? One we can call it as the troposphere, which is the first one. And the second one, what we can call it as the stratosphere. Correct, right? So which one of the following gases is present in the stratosphere at higher concentration? So when compared to the troposphere, stratosphere is the one which contain the more ozone. Very simple, direct question. To be honest, NCERT question. To be very honest. Okay. This is also missed, friends. This question also I will solve it in the tomorrow class. I forgot to keep the graph. But we have studied this. We have studied in the population ecology. Very direct question. I'll keep it the graph tomorrow without fail. Two questions are missed. I will solve it in the tomorrow class. Consider the following ecosystems. See, they are asking among all which will contribute most NPP, not the GPP they asked, not the gross primary production. They have asked net primary production. Very good, Abdul. This is a direct question, friends. I don't need to explain this. When compared to the rainforest, open ocean, algal beds, coral reefs, marsh regions and swamp region, they will give the more amount of the NPP. Then followed by the second is open ocean, third is the tropical rainforest, fourth is the algal beds and the coral reefs. Okay, so the first one will be the marshes and swamps. Don't get confused with that, friends. Okay, good. No, the answer is D, marshes and swamps. Don't confuse. Oh yes, the increased order only, no, they said. Okay. Sorry. They have asked in increasing order. Sorry. Right. Algal beds and coral reefs. Correct. I'm so sorry. Sorry. Answer C is the correct. Sorry. I did not see the increasing order. Correct. C is correct. Algal beds and coral reefs. Right. Correct. Okay. Historical frequencies of a fires in an area can be determined by. So sorry. See, actually, dear, I did not given the order. Okay, I did not keep the options. But what I am saying is, tell me among all which will be the increasing order. I understand, Tanvi Saima. I did not given that order options. I was asking directly which is in the increasing order. Whatever you said is right. Answer C is correct. That is correct. I did not given the 4 different options. Instead of asking that, you have to tell me the increasing order, which means you have to choose one option. From there, the ecosystem productivity is getting increased. Don't get confused. You have to choose one option from where the increasing productivity is increasing. Increasing NPP. You have to choose from the least one. Obviously, the algal beds and coral reefs. That is right. This question will be the different question. I kept the answer for the question number 56 and 57 confusiously. Don't confuse. The answer for the question number 57, I am telling you C. That is right. Don't get confused. That is right. Then when it comes to this, then it will be CBAD. Correct. CBAD. Don't confuse. Right. Okay. Historical frequencies of fires in an area can be determined by. This is called an analytical question. For this, you have to think it. How can we determine the fires, the probability of occurring the fire in that particular area is more? Friends, they have asked historical frequency. You know, here the word historical frequency will make us confuse. Here the word historical frequency means quantitative approach. Don't get confused. Historical frequency or quantitative approach is same. So what? Obviously by using the fire scares or tree ring analysis. By using the fire scare or the tree ring analysis, we can determine easily whether the probability of occurring the fire is in that particular area is possible or not. Option B will be the correct. Perfect. No, Sumaya, we don't consider the carbon content. Carbon content is no. You tell me if the carbon content is more or less. How can we determine the fire over there? Carbon content is not the correct one. No, 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 no. It cannot be a carbon content. The answer will be the B. Friends, being with the help of a measuring carbon content, we can check the photosynthetic efficiency of the plant in that particular area. Okay, and based on the carbon content, you can also tell the growth rate. You can also tell the growth rate, but you can't completely determine. That's what they have used a word historical frequency, which is a quantitative approach. Carbon content is not the sole source to explain whether the fire is occurring in that particular area or not. This cannot be a suitable measurement. The correct one is the, remember friends, tree ring analysis method. It's a newer method. It is the quantitative approach which is help us to understand the frequency of a fire. Don't get confused. If you want, you search in the Google, you can understand it. Okay, there are 10 more questions are there left, friends, that we will finish it in the tomorrow class, and in the tomorrow class we will move on to the part C. Okay, don't get confused, don't get confused. Okay, so friends, I'll see you at 3 p.m. We will continue the enzyme kinetics. Okay. Okay friends, thank you all. I'll see you in the afternoon 3 p.m. class. So without wasting the time, please revise it. Okay, and whomever did not take the mock test, take it at least today. By tomorrow another mock test will be uploaded. That is the second mock test. Okay, so whomever did not take the first mock test, please take it without fail. Okay friends, thank you all. I'll see you in the afternoon class. Bye bye.